stem cells. There have been a lot of talks about stem cells during medical week, so my main focus is going to be on the kidneys and the function of the kidneys. But I will be talking about stem cells later on in my presentation. So first I'm going to look at the main function of the kidneys. And the main function of the kidneys is just to regulate whole body homeostasis. And this is mainly done through the reabsorption and secretion and excretion of products. However, the kidneys do secrete some products, which is something that I never knew. So I'm going to be talking about that as well. So firstly, the kidneys go through ultrafiltration. And this essentially happens at the renal corpuscle, which is a name ascribed to both the Bowman's capsule and the glomerulus. The glomerulus is like a collection of capillaries, and it's situated at the beginning of the nephron. And the Bowman's capsule collects the filtrate from the glomerulus, and it surrounds the glomerulus in order to be able to do so. But the glomerulus is very unique in that it receives its blood supply from an afferent arteriole and drains into an efferent arteriole rather than a venial. And this creates high pressure because the arterioles both have a high pressure, and this is what is required for the ultrafiltration process to actually occur. Now the glomerulus is designed such that small molecules such as glucose or amino acids are forced into the filtrate. However, large molecules such as red blood cells or proteins remain within the capillary bed. And this means that if those large molecules are found in the urine and hence are excreted, it could be an indication of declining renal function, which may have the underlying cause of hypertension or just high blood pressure. Now, the reabsorption process mainly occurs in the loop of Henle, but it also occurs in the proximal convoluted tubule, which is just that sort of collection of yellow things over there, and the collecting duct, which is like the long yellow line. And as you can see, the kidneys regulate many different solutes. However, I'm going to be focusing on some of the more main ones and some of the more obscure. So firstly, we have glucose. Now the kidneys have adapted such that all the glucose that enters the filtrate is reabsorbed. Hence, if any is excreted, it could be an indication of declining renal function. It could be an indication of diabetes mellitus, which is essentially just diabetes, as it means that there are very high levels of glucose in the blood that aren't being reabsorbed. And high levels of glucose in the blood is known as glucosuria. Now, kidney reabsorption takes place in the proximal convoluted tubule, so what's highlighted on the board over there, and occurs through a variety of processes. Firstly, glucose is co-transported into the cell with sodium ions, and this is just through the apical cell membrane, so the membrane facing the lumen. And the sodium ions and glucose ions, once in the cell, then simply diffuse along the concentration gradient. The glucose uses facilitated diffusion to do so. And the constant flow of blood through the capillaries surrounding the nephron maintains this concentration gradient. And a sodium-potassium pump is also located on the basolateral membrane, so the membrane facing the capillaries, in order to replace the glucose that's been, the sodium that's been reabsorbed. And it filters out that sodium, which maintains a concentration gradient for the glucose to flow down, as sodium will want to flow into the cell, and so the glucose will flow along with it. Another common electrolyte is water, and scientifically, the regulation of water levels is known as plasma osmolality, which is just a measure of the body's electrolyte to water balance. And this is detected by the hypothalamus, which is at the front of the brain. And the hypothalamus controls the pituitary gland, which is involved in all homeostatic functions. Now, most important concerning plasma osmolality is the posterior pituitary gland, which is composed of blood vessel connections from the hypothalamus, meaning it can respond to the chemicals that the hypothalamus says the posterior pituitary gland should be secreting. So let's say, for example, you're dehydrated. So plasma osmolality is high, and there are more electrolytes than there is water in the body. The posterior pituitary gland secretes antidiuretic hormone, or ADH, which makes nephron walls more permeable to water, and hence makes you more hydrated. However, in reality, it's more complicated than simply making nephron walls more permeable. ADH binds to specific receptors on principal cells in the collecting duct, which translocates aquaporins to the apical membrane, so the membrane facing the lumen. Essentially what this means is that by ADH binding to specific receptors on the basolateral membrane, it moves intrinsic proteins, so proteins which embed themselves in the cell membrane. It moves them to the apical cell membrane, and this allows the membrane to become permeable to water, as usually it's impermeable. Now there are two types of receptors that the ADH binds to, and they're known as vasopressor receptors, as they promote the vasopressor action. And one of them is V1, which is located on the capillaries, and this causes vasoconstriction, which limits blood flow to the capillaries, and so hence water will flow into the capillaries. V2 receptors are located on the basolateral membrane, so the membrane facing the capillaries, and they activate adenal cyclase, which is an enzyme which synthesizes cyclic AMP. And the synthesis of cyclic AMP causes a lot of specific events to happen, one of them being vesicles to move to the apical membrane and fuse with it. And these vesicles contain the aquaporins, which, as I said earlier, are intrinsic proteins allowing the apical membrane to become permeable to water. And water will flow into the cell using those aquaporins and flow into the capillaries through the basolateral membrane, which is freely permeable to water. Now, the osmotic gradient is set up by two solids, namely urea and sodium chloride. And essential for both, the apical membrane is not permeable to water, but is permeable to these two solutes. Therefore, when the aquaporins are inserted, water will want to flow into the cell through the osmotic gradient. And this is known as urea trapping and the Schindler effect. And those two solutes can later be excreted as necessary. This leads on to the excretion of urea. And urea is produced due to the metabolic breakdown of amino acids in the liver, and you see an amino acid on the board. And the metabolite of this is ammonia. Any ammonia that isn't converted into nitrogenous compounds through the biosynthesis process is converted into urea. Now this happens through the deamination process, wherein the amine group and the hydrogen atom of an amino acid react to form ammonia. And ammonia is toxic and so cannot be allowed to accumulate. Therefore, it reacts with carbon dioxide to produce a less toxic urea in water. However, urea is very harmful if exposed to for a long time, as it can lead to kidney stones. So its excretion is very important. As I mentioned earlier, urea simply diffuses along a concentration gradient. Closely linked to urea is uric acid. And uric acid is more harmful if exposed to for a long time, mainly because of its acidic nature. And it forms crystals known as monosodium urate, which, if deposited at the joints, can lead to gout. And this is a very painful condition. Uric acid is produced due to the metabolic breakdown of purines. And purines are essentially found in all DNA or RNA, and are found in high concentrations in high protein foods, namely the muscles, mackerel, and ironically the kidneys. And uric acid acts as an antioxidant to help prevent damage to the blood vessel linings. However, as I mentioned earlier, it can be very harmful if exposed to for a long time. Some animals possess the enzyme uricase, which essentially converts the useful acids of uric acid into more soluble components, known as allotonin. However, humans don't possess this enzyme, so uric acid is simply filtered out in the urine. So this leads on to the regulation of acids and alkalis in the body. And this is very important in the kidneys, as it's done in conjunction with the lungs, which excrete carbon dioxide. The kidneys, however, reabsorb bicarbonate. And generally, bicarbonate is reabsorbed rather than excreted, as the body should remain in slightly alkaline conditions of around 7.35 to 7.45 pH. Because there's a very narrow window of toleration within the body, the kidneys are important in actually excreting these acids, as without it the body could enter alkalosis or acidiosis, which is when there are highly alkaline acid levels in the body. And this is done through a series of processes. Okay, firstly, carbon dioxide and water react to make bicarbonate ions within the cell, and this is done through two stages. The water forms its constituent ions of a proton and a hydroxide ion, and the hydroxide ion goes on to react with carbon dioxide to produce bicarbonate. Now this is catalyzed by the enzyme carbonic anhydrase, again situated on the apical membrane. The proton is filtered out using a sodium-hydrogen antiporter, which is essentially when a sodium ion will flow into the cell and the proton will flow out of the cell. And the bicarbonate ion will then flow out of the cell using symports with a sodium ion or antiports with a chloride ion. And again, symports, they both flow in the same direction, antiport, the bicarbonate ions flow into the capillaries, the chloride ions flow into the cells. This is, however, is only the first stage. The proton in the filtrate reacts with bicarbonate ions in the filtrate, as they cannot cross the apical membrane directly. They then use the carbonic anhydrase enzyme to react to form carbon dioxide and water. And the carbon dioxide ions react with the hydroxide ions to form, again, bicarbonate. This bicarbonate is then excreted in the same way as before. However, the symporter and antiporter are both electrogenic, which means they transfer uneven charges across the cell. And this means that a sodium-potassium antiporter is also located on the basolateral cell membrane to regulate the differences in charges. Not only does it regulate this, it also sets up a sodium concentration gradient as sodium ions flow out of the cell to flow out. So this concludes the homeostatic functions of the kidneys. But now I'm going to briefly look at some of the substances that the kidneys actually secrete, because I thought that was quite interesting. Now, erythropoietin is a glycoprotein, and it controls erythropoiesis, so essentially the production of red blood cells. As you can see, it acts as a cytokine for erythrocyte precursors in the bone marrow, which essentially means that it's a signaling molecule for red blood cells found in the bone marrow. And it's only produced in the kidneys after birth. It's produced in the liver perinatally, and it's produced by a collection of cells that surround the kidneys. Next up we have renin, and renin is very important in the regulation of blood pressure. And in fact, it is an enzyme rather than a hormone, as it has no receptors on its surface. Renin is secreted by afferent arterioles from the juxtaglomerular apparatus in response to 3 different stimuli. A decrease in atrial blood pressure, and this is detected by baroreceptors, which are a type of mechanoreceptor, which means they're sensitive to blood pressure. And if the blood vessels are stretched, so there's vasodilation and blood pressure therefore decreases, the baroreceptors detect this and secrete renin, which relays a signal to the central nervous system. And this is the most direct link between renin secretion and blood pressure regulation. Renin is also produced in response to salt concentrations and the sympathetic nervous system, which controls all homeostatic functions. And it takes part in the renin-angiotensin system, which further controls blood pressure. So renin secretion within the kidney is actually very important in the control of blood pressure throughout the whole body. Lastly, I'm going to be talking about vitamin D, which is more accurately, the biologically active form of vitamin D, which is known as calcitriol. And this, again, is produced in the proximal convoluted tubule by mitochondrial oxygenases, so enzymes which add oxygen to substances found in the mitochondria. And this catalyzes the hydroxylation of calcifidiol, which means that it sort of adds OH groups, so it makes something more alcoholic, and calcifidiol is just essentially produced in the liver. And interestingly, vitamin D, calcitriol, controls calcium ion homeostasis, therefore it's increased by the production of prolactin, which essentially stimulates lactogenesis, so the formation of milk in the mammary glands. So why am I telling you all this? It might have seemed like a lot of information to just throw at you, but I think it's very important to grasp how influential the kidneys are within the body, because chronic kidney disease is described as a progressive loss of renal function for over 3 months. And although it comes in stages of severity, it has no known cause, nor does it have a known cure. And that's why chronic kidney disease is really a problem, because patients either have to await a kidney transplant, which can take a very long time, as you probably know, or they have to rely on dialysis, which is a very long and grueling process, because you have to go to the hospital for hours a day, days a week. In fact, it causes 45,000 premature deaths in England alone, and that's more than breast and lung cancer combined, so it's not a small figure. And more worryingly, I think, is the fact that it's a predicate to cardiovascular disease. So if you do contract CKD, you're unlikely to die of CKD itself, but the complications that arise with that, so uric acid buildup, urea buildup, no regulation of blood pressure due to the secretion of renin. And you can see that there are actually a lot of people, this is in England alone, who are affected with this disease. And because it has no known cause, it can seem like quite a bleak situation. But here, potentially, stem cells could come to the rescue. And as you probably already know, stem cells is essentially the term defined to undifferentiated cells that are able to specialize into many different cell types. And there are 3 types of stem cells. Embryonic stem cells, found in the embryo, and they're pluripotent. And what that means is that they can form any type of cell in the body, minus the extra embryonic cells, so the placenta cells. And cells that have that ability are known as totipotent. Umbilical cord blood stem cells are found in the umbilical cord, obviously, and they have features of both pluripotency and multipotency. Now, multipotent stem cells are cells that can differentiate into many different cell types. However, they're restricted to a particular physiology or organ or tissue type. And adult stem cells found in adult tissue are fully multipotent. So as I mentioned earlier, embryonic stem cells are formed in the embryo, more accurately the blastocyst, which is a form of the early embryo. and they're found in the inner mass, and that's only composed of around 20 cells. So around 20% of the blastocyst actually has those stem cells. Umbilical cord stem cells are found in the Hutchins jelly, which is essentially a gelatinous connective tissue within the umbilical cord. And as I said, there's somewhat of a confusion because technically they're pluripotent, but when they go on to divide, they don't do so as well as you'd expect them to. Having said that, they have a plethora of advantages because they're readily available since they're produced with every birth. There's no ethical conundrum as you have with embryonic stem cells, and this has actually led to limited funding within research within embryonic stem cells. They're very plentiful, which means that not only can research into them be more extensive, but any treatments developed by them can be widely distributed. The actual cells within the cord blood have very limited development of antigens, which means that they can be transplanted into the body without detection, and so without injection. And they have a very underdeveloped immune system, the actual cord blood, which means that you avoid graft versus host rejection, which is when the actual transplanted tissue attacks the body's own cells. Now, adult stem cells are quite hard to isolate because those stem cells tend to sort of blend into the body's own cells. However, Oliver et al. Did a study wherein they found stem cell niches within the kidneys in the areas highlighted in green, but the most promising results were found in blue. Having said this, these niches don't actually contain that many stem cells, although it's more than you can find just lying around in the body. In addition to this, these stem cell niches, the stem cells found in these niches, have a very limited capacity to grow in vitro, so to grow in a lab, which means it can be very hard to study them, and yeah, it's just a lot of complication really with adult stem cells. And in addition to this, Davis et al. Did a study wherein they found that it's not only the chemicals that the stem cells are exposed to that causes them to differentiate, but also the environmental and physical factors. And this is important because you could isolate those stem cells from those niches that Oliver et al. Believe they found. You could then grow them in a kidney, in the lab, by exposing them to the correct chemicals and to the correct sort of environment. And then from this, you could actually grow your own kidney, technically. You use the extracellular matrix of the kidney. So you've got the sort of scaffolding of the kidney, and you take away the cells, that's the extracellular matrix. You take the stem cells from the kidneys, you cause them to differentiate by exposing them to the right conditions, and you sort of plant them on that scaffold and grow your own kidney, which is especially tailored to that person, and so avoids rejection completely because you're using them themselves. And I think that is very, very interesting. So how do stem cells differentiate? I think if you're able to actually understand how they differentiate, then you can grow them in the kidney in the lab, knowing how they'll differentiate, and then transplant the mature cells into the body. Or alternatively, you could transplant the stem cells into the kidney, knowing full well how it will differentiate, and then just reap the rewards really. Eisenberg wrote an article wherein he found instances of transdifferentiation. And this is when multipotent stem cells differentiate into stem cells that aren't of their expected lineage. However, this was quite a sporadic process, and so it's not very helpful for CKD sufferers. Neither is reprogramming, which is essentially when you take mature cells and you get them to differentiate into different types of mature cells within the same lineage. But I think most sort of interesting for CKD sufferers is induced pluripotency and iPSC, so induced pluripotent stem cells. And this is when you take a fully mature stem cell, you add an inducing factor, and you cause that cell to revert back to its embryonic stem cell-like state by forcing it to express certain genes and factors important for maintaining the defining properties of embryonic stem cells. And by doing this, you're able to actually get stem cells from that person's own body and use it to then grow the kidneys and transplant into them, etc., etc. However, although these seem similar to embryonic stem cells, it can't be said that they're the same, and that uncertainty obviously has to be researched further before they can actually use in treatment. And in addition to this, when done with animal studies, they induce the pluripotency using a virus, and this has led to cancers, and obviously you don't want to give someone already suffering from chronic kidney disease cancer. So that's something that obviously needs to be researched further as well. But I think this is definitely very, very possible and potential. In fact, scientists in a Harvard laboratory have actually been able to grow a fetal kidney using the very same iPSCs I just talked about and embryonic stem cells. So this isn't something, sort of out of Frankenstein's lab or anything. It's something that's actually being done and something that could be actually very useful in the future. So in summary, I'd just like to say that although not sort of appreciated mainstream, the kidneys are very, very important in that they control the whole body. And that's why chronic kidney disease is such a problem, because not only does it lead to such a debilitating disease wherein you're left with only 10% renal function, and that is really, really bad. You can practically move with that kind of renal function. We don't know what causes chronic kidney disease, and we don't know how to cure chronic kidney disease. And it's a disease that not only will affect the patient, but will affect families as well, because if you have someone going on for hours at a time, days on end, on dialysis, it's going to affect home life. And that's why stem cells could be so important, in that they can be used for treatment when you're growing a kidney, but they could be also used for research into what causes chronic kidney disease and how to treat it. So I'd just like to thank you all for listening, and if you have any questions, please.
You talked about how there's no cure for renal failure. Do you know how the symptoms of renal failure is treated?
Well,
besides from dialysis.
Yeah. Generally, a renal failure, particularly in the early stages, is asymptomatic. So people who actually are diagnosed with chronic kidney disease get that diagnosis through presenting with heart problems, and then it's found out that the chronic kidney disease is what caused heart problems. One of the symptoms I know is uremia, which is when you have sort of like a white sort of substance, which is the urea being excreted from your head. And generally, I don't think you can really cure that rather than regulating the
Yes. You may have, when we got to the first part of the presentation, you may have just already been shown my head, but is it because of the processes of secretion that this tubing in the kidneys?
Oh, that's really interesting. Okay, basically, there's like an adrenal gland located on the kidneys, and each adrenal gland is differently shaped. And I don't actually know how that affects the actual renal function, but each kidney is slightly different, but both kidneys will excrete and regulate the same sorts of electrolytes. But obviously, if you have two, teamwork better than
when you said that they study how they want to study how stem cells different and they take it outside the body and they grow it outside the body until the stem cells are mature enough. Do you know like the process of how they get it to the specific place in the body to then grow into a kidney? What process do they use to inject the stem cells into the body to get it to a specific place?
Oh, I'm not sure actually. I mean, you'd have to localize it definitely, and because it's in its infancy stages, I don't actually know how you'd be able to just inject specific types of cells into the kidneys. But I'm assuming it would be an injection, or alternatively they could do an open surgery and transplant those cells in. But I'm not sure.
You said there are no known causes for the disease. Are there any links to like lifestyle?
Yeah. There are no known causes, but there are risk factors. So if you have high blood pressure, I said that the glomerulus is supplied by two different arteries essentially. If you have high blood pressure, that glomerulus is going to be working under very high pressure, and that can lead to failure. One cause that's been suggested is vasculitis, which is like inflammation of the capillaries. And obviously, because the tubule is surrounded by blood capillaries as it regulates those substances. If those are inflamed, then it's going to put pressure on the nephrons. So that's also a known cause. But in terms of lifestyle, high salt intake can also have a play factor. I think genetically, if you are Caucasian, then you're less likely to get chronic kidney disease. And yeah, I think that's about it. But obviously a healthy lifestyle is going to help.
Given that the use of stem cells in medical research is a relatively new development, how much research has already been done into sort of preventing chronic kidney disease, and how reliable is stem cell research, given that it's so new?
Well, I think in terms of stem cell research being new, I think it's quite new, but it was done sort of hush-hush prior to the 21st century, and then it became very public in around 2002 when it was like, oh, stem cells cause cancer and all that kind of hype. Then it sort of faded into the background again, and then a lot of research was done in like inducing pregnancy, that kind of thing, which is, I don't know if you went to any of the medical talks, but one of the guys who was doing it said he was doing research into it at the time now. So it's coming back to the forefront. But in terms of actual using the stem cells for research, I think it's important to use stem cells for research because if you know how those cells differentiate, you can then see why it's differentiating into the cell that's causing the chronic kidney disease. And from there you can isolate a cause, you can find out what kind of treatment is effective. I mean, that's for cells in general, but I think with stem cells you can actually see the process of maturation, and I think that's important as well.
Do you know what results in kidney pain? Like, people often present with symptoms of having like pain in their back around their kidneys, or like pain in the kidneys. Do you know if that's do you know what that is?
It could be kidney stones.
Okay.
Maybe if you fell badly and your kidneys are bruised. But if you're presenting back pain, it doesn't necessarily have to be the kidneys.
Oh no, I meant like if you're presenting kidney pain.
Yeah.
Like, do you know what causes kidney pain?
Not really. My best guess would probably be kidney stones, because kidney stones are found within the kidney, and that's when you're putting pressure